here to talk to you today about the structure of DNA. So please make sure you fill out your Cornell notes as you're going through this. Pause when you need to and then move on. So discovering the structure of DNA. Even though Griffith and Avery, Hershey, and Chase provided the evidence that DNA is a carrier of genetic information, at this point we still did not know what DNA looked like. If you need to pause, go ahead and do so you can finish writing this slide. Continue on in that section, James Watson and Francis Crick were given all the credit to finding the structure of DNA. Maurice Wilkins allowed Watson and Crick to see Rosalind Franklin's work without her permission. So they were basically lying and cheating to get credit for discovering the structure of DNA. Go ahead and pause if you need to and finish the slide. Rosalind Franklin is also known for taking a photograph of DNA and then doing year-long calculations to determine the actual shape of DNA. She died without knowing Watson and Crick saw her work and has not received credit for her work. Go ahead and pause if you need to and finish the slide. Watson and Crick took a shortcut and made several models based on the picture Franklin took. Eventually, they came to the correct conclusion, and DNA takes the shape of the double helix. So now we are talking about the shape of DNA. This section is towards the bottom of your front page. If you need a moment, go ahead and pause and finish writing the slide. Going on to the next section labeled the double helix, it says a double helix is made of two strands that are twisted together. It looks like a spiral staircase. So for example, these blue lines would be like your handles going down the stairs, and the lines in between would be like your steps. It also kind of looks like a twisted ladder. Go ahead and pause if you need to finish writing. DNA is made up of nucleotides. So now we're talking about the parts of DNA. Nucleotides are a type of macromolecule that we talked about during the first 9 weeks. Nucleotides are the monomers, or basically the building blocks of DNA, which is a polymer. Polymer means that it's made up of more than one monomer, or more than one nucleotide in this case. What I'd like for you to do in your notes, you'll notice there's a little section off to the side that asks you to draw this picture. Please draw this and label this picture off to the side. The orange circle represents the phosphate group that helps make up the DNA molecule. This pentagon here is representing the deoxyribose sugar. That's how we get the deoxyribonucleic acid name. And then we have a base off to the side, which we'll talk about in a second. Go ahead and pause if you need to finish the slide. So we're still in the parts of the DNA, and we're going to talk about the sides or the backbone. The sides of the backbone of the DNA are made of phosphates and sugar, or specifically deoxyribose, that alternate down the side. So basically down the side, if it starts off with a phosphate, it'll go from phosphate, deoxyribose, to phosphate, to deoxyribose. And continue on until it ends. I have some pictures I'll show you later, and we'll practice that together. Attached to each sugar is one of the 4 bases. Go ahead and pause and finish writing if you need to. The bases are adenine, which can be represented by A, cytosine, which can be represented by C, guanine, G, or thymine, T. Go ahead and pause if you need to finish writing. So the bases are split up into 2 groups. We have purines, which are made up of adenine and guanine in 2 rings. Pyridamines, which are cytosine, and thymine, which is made up of 1 ring. And I'll show you what I mean in a second. Go ahead and pause if you need to, and then continue when you're ready. Purines. Purines have 2 rings attached to each other. So we have a pentagon and a hexagon for adenine and guanine. For pyrimidines, which are cytosine and thymine, we only have a hexagon. Okay, notice here's your phosphate group, and then it goes to deoxyribose, phosphate, deoxyribose, phosphate, so on down the line. Notice that your deoxyribose is attached to that base. Now we're on to base pairing, which is a subsection underneath parts of DNA. In order to make up the middle of the DNA, the bases need to pair up. A pyrimidine pairs with a purine, and they form hydrogen bonds between the pairs. Go ahead and pause if you need to. Adenine always pairs with thymine, and there are 3 hydrogen bonds between them. Guanine always pairs with cytosine. There are 2 hydrogen bonds between them. No exceptions. Go ahead and pause if you need to and continue when you're ready. So an easy way to remember is to use the abbreviations. A pairs with T, which spells at, and G pairs with C, which is not a real word, and CG is not a real word either. So that's how I keep it straight. Pause if you need to, and move on when you're ready. So now it's time to practice. We're going to write the base pairs that complement the following half of the DNA. So the black strand up here at the top is my original DNA sequence. The red strand is my complementary strand. Notice my first letter is A. A always pairs with T, or thymine, so I write T below it. Cytosine always pairs with guanine, so I write G below it. Then I have G on top, so I'm going to write C below it because guanine always pairs with cytosine. I have another cytosine, so it will pair with guanine. Another cytosine will pair with guanine. Guanine will pair with cytosine. Guanine will pair with cytosine. Adenine will pair with thymine. Thymine will pair with adenine. Guanine will pair with cytosine. Adenine with thymine. Cytosine with guanine. Guanine with cytosine. Guanine with cytosine. Guanine with cytosine. Adenine to thymine. Adenine to thymine. Thymine to adenine. Pretty simple and straightforward. Now you go ahead and try and write the complementary pair to this strand. This is a picture of a flattened out piece of DNA. Notice we have our phosphate group at the top, followed by our deoxyribose, which is attached to cytosine. If you look on the other side, our deoxyribose is connected to a guanine. These red dots in between these two bases represent the 2 hydrogen bonds that are formed. If you look at thymine and adenine, there are 3 hydrogen bonds that are formed. You should be able to label something like this. The circles represent phosphate groups, the pentagons, deoxyribose, and then depending on how things are laid out, you will be able to label cytosine, guanine, thymine, and adenine. You should also be able to label that they have 2 double bonds or 3 double bonds depending on which pairs are pairing up. That is all for today, guys. I hope you guys have a great day, and let me know if you have any questions.